this amazing place. And I'd like to start with a very personal note. I was, just a few months ago, I had the chance to be at St. Thomas's Abbey in Brünn, the Czech Republic, and the occasion was Gregor Mendel's 200th birthday. I was allowed to speak there, which I regard as a huge honor, and I took these pictures on his birthday, actually. That's the bedroom and study room in this Augustinian monastery. And there's quite some relevance to this meeting in this, because Gregor Mendel is not, in my mind, not only the founder of genetics, but also the founder of data science. He draws conclusions from growing over 30,000 pea plants, and this was, of course, facilitated and enabled by the Augustinian monastery. And today I feel I'm having an even greater honor to be able to speak here. And I look forward to the scientific exchange with the Pontifical Academy of Sciences here. Going further towards what I'm actually going to present to you about, as Edith mentioned, I'm EMBL's head of data science, and we recently started a data science team at the EMBL, given the huge growth in data we see with data size across different disciplines doubling every 18 months. Here symbolized by cancer genomes and also rare disease genomes, which actually in honor of Mendel are still called Mendelian diseases that are being studied in these settings. EMBL has a fairly unique setting. We are an institution with 6 sites, as Edith mentioned, and we actually have the full life cycle of data within one organization with these 6 sites, from the planning of an experiment to the generation of the data, to interpreting the data, to then publishing and archiving the data at the EBI, and then allowing a new cycle to take place, which for us has been a huge motivating factor in getting a data science team up and running. And as Edith also mentioned, there's a huge focus on open data within our organization. The new Data Science Center, which I'm heading, is there to enable both research and services to ensure that research data can be curated in an expert manner, annotated, managed, integrated, visualized, and importantly shared with individuals to lead to better research. And I'm going to briefly show you the priority areas for this new theme. The areas are on research methods, including methods in artificial intelligence, integrated research data management, to allow data to be used in a collaborative manner, biocomputational workflow sharing, to allow experimental groups to benefit from methods and use them in their everyday routine, and development of infrastructure solutions, expansions of embedded services, as well as, last but not least, a huge and important focus, the training of a new generation of data science. So how is this all linked to improving inequalities and tackling inequalities in cancer? And I'm going to be fairly broad here and going to repeat some principles that said previously, but emphasize what I think is really important. So with data science and artificial intelligence, we are able to integrate heterogeneous data. And this can actually improve clinical decision-making because we can integrate both unstructured data sets as well as structured data. With AI, there's the opportunity to go through tasks that are normally done by experts and perform classifications that might not be always as good as for the best experts, but are nearly getting there. An example here shown from my past EMBL work on breast cancer, where AI software can identify TP53 mutant and breast cancer and TP53 wild-type breast cancer. So this provides opportunities to be deployed in regions that have poor access to excellent pathologists, and therefore bring in more inequalities into the clinic. Potential additional future applications one could think of, in addition to cancer diagnosis, is to predict drug responses from images, to train doctors via remote training tools in areas where training is rare and poor. And even mobile chatbots with large language models such as ChatGPT could be utilized to help patients manage their treatment regimen. Lots of opportunities, but also caveats, as Olivier has pointed out. There's a challenge of AI containing biases, sometimes unknown biases, demonstrated here by software from the Fraunhofer Society to screen for skin cancer with lesions using AI. And of course, if these softwares are only trained with individuals that have a light skin, they may not work. at all or not work as well in people with different skin color. Another bias or caveat of these tools is that they're often acting as a black box, and specifically when we apply them clinically, we need to get beyond this, and there's active research in the AI field and also actually taking place at EMBL to get better insights into the basis of classification, to go beyond this black box. Also, the underlying basis of all of this, these artificial intelligence softwares are very data greedy, so we need to be able to share data. That is the basic principle of it all. And in this regard, essentially a decade ago, I co-initiated and since then co-led the pan-cancer project that Edith mentioned in her talk. We published our work in a series of papers in 2020, uncovering some general principles of cancer biology through it, and really demonstrating how data sharing is crucial to enable studies with cohorts. And now a decade later, it still concerns me that we have not achieved this mode of, or this global data sharing in the clinical sector. I know there's many challenges with it and good reasons to be protective about data, but it would be good to get there soon because patients will need it and there's possibilities emerging from it. And there are several efforts in Europe promoted by EMBL and others to get over this. So now I would also utilize the chance to speak about our own research for the remaining 8.5 minutes. And I'll start in a very basic manner to talk about cancer as a disease that is largely caused by somatic mutations that arise in a stepwise manner, a process that can take years or decades, which is akin to Charles Darwin's principle of gradualism and evolution at the cellular level. Interestingly, recent studies, though, have shown that numerous human tissues, actually virtually all of our tissues, acquire sequential mutations over life. And usually that happens without cancer developing. And therefore, it was recently proposed that this model may actually be incomplete, and there could be a rate-limiting process that distinguishes cancerous from non-cancerous cells, in spite of the stepwise acquisition model. And I believe an excellent candidate for this rate-limiting process are complex genomic rearrangements, because they can very rapidly remodel the karyotype in one or a few cell division cycles. Chromothripsis is one example of this. This is a process whereby chromosomes shatter, as indicated on the slide here, and then rejoin through aberrant repair, resulting in multiple alterations happening at the same time. My lab pioneered work on chromothripsis in a brain tumor termed medulloblastoma. We showed that a common outcome of chromothripsis are circular extrachromosomal DNAs, which undergo further recombinations, again rapid, to remodel the genome even further. We now know that these complex rearrangement processes are detectable in most cancers, and they're linked to poor prognosis, metastasis, and therapy failure, suggesting they actually may be key drivers of malignancy. But what does that mean to our understanding of cancer evolution? Well, if these rapid explosive processes are key drivers of malignancy, then we need to actually amend models of how cancers arise. In addition to the gradualism model of Charles Darwin applied to the cellular level, we need to invoke other models of evolution, such as saltatory evolution events, which are here defined as giant adaptive leaps, but giant selective leaps that happen in a very short, explosive genomic crisis. And this, intriguingly, could actually draw the line between a normal benign cell and a full-fledged cancer cell, because these changes may make a cell extremely evolvable, allow it to evade the immune system, to go into new tissues, go invasively in metastasis, as well as relapse. But studying these events is actually tricky, and although this concept may be plausible to actually study complex rearrangement process, saltatory evolution, is extremely challenging. The reason for this is that we typically, in cancer genomics, analyze snapshots of mutations long after the events have taken place, often years later, once surgery has been carried out in a patient. And hence, we do not know several basic principles, such as how common complex rearrangements are in cancer evolution, in what context they arise, and which mechanisms operate, and at what rate. Again, when we look later, once the tumor is surgically removed, we can make guesses, but we will not be able to uncover these parameters. And there's some key methodological needs and current challenges that we set ourselves in the lab. Actually, we set ourselves a fairly high bar, and this I'm now summarizing work of several years in our lab we wanted to have a technology that allows us to detect different forms of structural rearrangement or structure variation at a very wide clonal frequency range, along with a functional readout. And current mainstream technologies don't provide for that. I'm going to go through them one-on-one. So if we sequence genomes in bulk from bulk tissue, we will average characteristics across cells and tissues and are insensitive towards low-frequency rearrangement. We could also pick cells and grow them to clones and study them by deep sequencing. This has been done and actually gives access to wider clonal frequency range, but it's restricted to culturable cells. And those with complex rearrangements, we believe, are probably often not culturable. So it's also not really applicable. Single-cell analysis, of course, is promising, gives access to the widest clonal frequency spectrum, but methods that utilize mainstream whole genome amplification will miss complex and balanced rearrangements because of the artifacts that are introduced by these methods and because they're really low coverage and will not be given us detail at the mechanistic level. They will be given us copy number profiles from which we, however, cannot reconstruct mechanisms. So we switched gears, actually, and repurposed a technology originally developed in Peter Landstrup's lab, and we reconfigured it, which now gives us access to DNA rearrangement process and complex rearrangement processes as they occur in single cells. The way this method works, we're incorporating dual DNA replication, a marker, DRDU, that allows us to sequence the genome of single cells in a homolog and strand directional manner. So each chromosome is sequenced on a single strand. And we developed computational principles in our lab to mine these data and reconstruct karyotypic rearrangements from them at high resolution. This method is applicable to primary samples, organoids, and so on, and it's pretty scalable. Just to show you how this principle works, we're looking at 3 different readouts in each cell. How many reads are mapping to a certain interval, the depth, which strand direction they have, and which homolog phase they're coming from. And the combination of these 3 features gives us every rearrangement class that is present in a cancer genome, so allowing us now to identify chromothripsis events or other events that happen and rapidly reconfigure a cancer genome, such as breakage fusion bridge cycles. We're also seeing copy number balanced events, such as translocations, all with a single technology. Recently, we realized that in the very same cell, we can also gain a functional readout, and that is by zooming into the data very closely. Because micrococcal nuclease is used to construct strand-seq libraries, we get a perfect readout of where nucleosomes sit in each cell. You can see how this aligns with MNA-seq data, giving us the same position spec, where the strand-seq data is unique because the data is resolved by homolog. So we get homolog-resolved nucleosome occupancy data, which, using deep learning, we can convert to a gene expression readout, because genes that are actively transcribed miss nucleosomes at their start and end site, and also throughout the gene body. And we showed that we can, for genes that change in expression very vastly, the top 10 dysregulated genes, we catch 9 with this method. Again, in the same cell in which we get a DNA rearrangement readout, allowing us now to bridge between complex rearrangement processes that emerge in single cells and their function. And as a proof of principle, we recently applied this to primary cells from leukemic patients. This is here shown a TLL, a childhood TLL, which showed a subclonal chromothripsis event that we could verify by bulk sequencing, going extremely deep because present only in a sub-portion of the cells. And with a method, we could predict the actual functional consequence of this chromothripsis event. In this case, we infer that the MIP oncogene is overactivated along with its downstream targets, which includes Notch, a targetable gene, allowing us to assess, in this case, not in a clinical setting, in vitro setting, whether we can act onto this phenotypic readout. And indeed, with a pan-Notch inhibitor, we see reduction of the chromotryptic clone and not of the wild type. So showing we can now go from complex rearrangement processes to a functional readout that is phenotypically targetable. So my last slide, since I have 30 seconds back, I can only tell you we're currently expanding this significantly together with Andreas Trump in a primary leukemic setting in complex karyotype AML, where we are uncovering amazing patterns of structural abnormality and ongoing heterogeneity with consistently viewable subclones that appear to change their character virtually from cell to cell. I'm showing here a quick highlight of this with a particular pattern that we attributed computationally to a circular chromosome that was then verified to be karyotypically really a circular chromosome, and a suggestion for a mechanism that leads to ongoing karyotype heterogeneity, potentially driving relapse. And with this, I thank you for your attention.